To welcome you today to the 2024 Immcantation Users Group meeting. As many of you are hopefully already familiar, Immcantation is a start-to-finish analytical ecosystem for adaptive immune receptor repertoire sequencing data analysis, or what we call AirSeq data analysis. Hopefully that term's getting more familiar to everyone. So we're really excited that Immcantation turns 10 years old this year. The first two papers that form the basis of the framework, Presto and Changeo, our first two packages, were published back in 2014. So that makes 10 years since those first publications, which sought to standardize the file formats and some of the analysis for air sequencing data. Quite a bit has changed since that time. When we started out, when AirSeq started out in about 2009, every lab had different experimental protocols for generating these types of sequencing data. We generally were not getting full-length VDJ. We had primers in the V genes. We were generally doing bulk sequencing and only getting heavy chains, unpaired with the light chains. And now the landscape has changed quite a bit. I think as many of you are undoubtedly aware, we have several companies where you can buy kits for doing standardized air sequencing data generation. Most of the time, we're getting full-length VDJ with constant regions included. And more we're seeing single-cell experiments done where you get not only you get the paired heavy and light chains together, often associated with the transcriptome of that single cell. And we're starting to see more AirSeq incorporated into spatial RNA sequencing. So we're really excited about those trends. We also have a more organized community. For those of you who are not familiar, we have the AIR community, the Adaptive Immune Receptor Repertoire community, which is part of the Antibody Society, which has working groups and an annual meeting, which we'll tell you a little bit more about at the end of the users group meeting today. Before I hand things over, I just really wanted to thank all of you, the users of the Immcantation framework. Really appreciate that you make use of these tools and the feedback that you give us. We really depend on a lot of feedback, both for feature requests and interface fixes and things like that. Also, really wanted to thank everybody who submitted abstracts for the users group meeting. Unfortunately, we did not have time to include everyone, but we're hoping to offer this again next year. And finally, really wanted to thank Susanna for really making sure this users group meeting happened, along with Gisela and Adele, and you'll hear from all three of them over the course of this users group meeting. And of course, our funding from NIH, BSF, and Pharma Foundation, mainly for the toolkit. We're really excited about the next 10 years, and with that, I will hand things over to Susanna.
So as Steve said, Immcantation has turned 10 years, and part of the success and how we could make it to 10 years is because of the adoption of the tool and because we have such a fantastic user base. The figure on the left side, the red numbers, are the numbers from today's meeting. Close to 200 people registered for this meeting. Most of them are in academia, and some of them are in industry, and the small amount belong to other areas of work. And they left us some messages. Some are very encouraging messages, like keep up the good work. I must say, I know this person. He was a lab member, and it's fantastic to keep in touch with people that we used to work with in Immcantation. Some other messages are for people that are maybe starting to learn to analyze single-cell data and hope to learn something from this meeting. Others want to know how other people are using Immcantation. We also have people that are working in other software tools that want to learn how we do things, which is also very nice because that shows that they value our work and that we are a reference for the community. Super nice as well. And the numbers on the right show the numbers of visitors that we have to the website and the number of downloads. And since we started tracking at the end of 2016, we see that we have a constant number of downloads, and we are growing. So it seems that the tools are really useful for the community and they are using them. As you know, Immcantation is developed in the Kleinstein lab at Yale University, and it's developed in the context of research projects granted through funds, funded through grants from NIH and other sources, as Steve mentioned. And behind Immcantation, there is really a lot of work, the work of many people. There is the Immcantation core team that are the people that are contributing code, but there is also the extended Immcantation team, which are other lab members that use the tool and provide feedback. They discover things in their data that we need new methods, new filters. Maybe something is not clear. They help to create tutorials. They help improve the documentation. We also have lots of collaborators that generate data, some other computational collaborators. So it's not just our work, it's the work of a lot of people. And as Steve mentioned, Immcantation is a start to finish analytical ecosystem. It's made of R and Python packages that are open source, and they are modular, and they are really designed for bioinformaticians, so they can build their diverse combinations of workflows or analysis using their expertise in data analysis. This is fantastic because it is modularity at great flexibility. It's also challenging for people that are new to the field because they need to learn how the different modules connect with each other, how they should use them. And to help with that, we have extensive documentation. We have done webinars, we have tutorials. We have an email that you can reach to us if you have any type of questions, not from installations only. You can also ask about how would you analyze this data. You can reach to us. And there's also the issue with having many tools that need to be installed, which is also a challenge for some people. So we have a Docker container that has everything ready to be used, and the code is a Bitbucket. And how the different tools communicate with each other is because they use the data standard developed by the R community that Steve mentioned. And that makes that you can use some of the tools, you don't need to use all of them, and you can move your data between the different tools. And also because it's using the standard, you can also connect to other tools that also use the same standard, third-party tool. And we released today, don't try to download it yet because I am not sure Docker has finished building the container, but we have released today 4.5.0. It has some updates. We have updated packages with bug fixes, performance issues. There may be some changes that could be breaking backwards compatibility, because I believe we added some additional arguments. If you have any issues, you can reach us to us by email, and we can help you figure out what happened. We also have revamped our tutorials. They are now in Markdown, Markdown format, because most of the framework is in Markdown. So you have one tutorial that introduces two analysis of visa repertoires, another one focused on single cell, and there is another one integrating VCR repertoire analysis with gene expression data, with single cell data. In the future, we want to continue to improve and expand the single cell methods. We want to continue to have tutorials. We want to improve the documentation. We want to facilitate the use of the tools in complex workflows, in integrated workflows. We have developed an Airflow workflow that Gisela is going to introduce you now. And we are very excited about this project. And we also want to develop support for spatial analysis. And for that, we're also looking for collaborators. So if you're working on spatial analysis and would like to collaborate, just send an email to Steve and we'll see. And now Gisela will tell you about Airflow.
Yes, thank you, Susanna. I will briefly introduce Airflow, which is this workflow that was just open in a preprint last week. So we're very excited about this work. And basically, Airflow is a best practices workflow for the analysis of bulk and single cell air-seq data using all of the Immcantation framework tools, or most of them, as there are many. And it's capable of analyzing both bulk and single cell data. So when it's analyzing bulk data, it can handle raw FASTQ files, and then it performs QC and sequence assembly with Presto, BDJ assignment and filtering with Changeo and IGBlast, clonal analysis and germline assignment with Scoper, Shazam, and Dowser, and lineage reconstruction with Dowser. And throughout all these steps, it also provides some useful QC reports and repertoire analysis reports that are produced with Enchanter, which is a new package that is now part also of the Immcantation framework. And for the single cell and assembled data coming from bulk sequencing as well, it's also possible to analyze this data with the Airflow workflow, but in this case, the workflow starts directly with the BDJ assignment. So I don't want to enter too much into details, but I want to just explain a little bit how it works. So the workflow is written with Nextflow, which is a workflow management system, and it has a number of features that makes this workflow portable, as Nextflow supports a variety of computing infrastructures, from running it locally in your personal computers to high-performance computing clusters and even cloud infrastructures. It makes it easy to have reproducible results, because Nextflow supports natively some container engines such as Docker and Singularity that contain all of the tools needed for the analysis. And because of Nextflow continuous checkpoints and implicit parallelism, it's possible to resume the pipeline whenever a process fails. And it's also possible to process samples in parallels, making it scalable to hundreds of samples. And the workflow is also part of the NF Core project, which collects community-curated open source Nextflow workflows for different bioinformatics analysis tasks. And as part of the NF Core project, the workflow comes with additional features, which are stable releases, which are also provided as DOIs to Zenodo, extensive documentation on the NF Core side, continued integration tests on commits and releases, and community code review that ensures that the code is good quality, reviewed with other community members, and that is accepted by the whole bioinformatics community, or like the one that is part of NF Core at least. So how to use NF Core Airflow? As a prerequisite, there's just Java installation, Nextflow installation, and the installation of the container engine of your choice, being it Docker, Singularity, or the container engine, or any of the other container engines that are supported by NF Core workflows. And then it can be just provided like as a command in the command line, Nextflow run, the name of the workflow and the latest release. And Nextflow already directly pulls the pipeline code for you, and also the containers needed for the analysis, so you don't need to take care of anything else. For analyzing multiple samples, it's possible to provide a sample sheet. Here is an example in which for analyzing single cell data. Here an example of metadata fields that are needed for the analysis, for example, sample ID, subject ID, file name. And in the case of single cell analysis, the direct output from Cell Ranger is supported in the array arrangement format, but also assembled sequences in FASTA format are supported. These are very few details I wanted to mention, just also to mention that we are already working on some additional features, which is, for example, providing preset parameters for common sequencing kits, for example, NEB. This would allow to just provide a parameter called -profile NEB, and that will already give some preset of parameters. And we are also already working on including TRUST4 into the workflow to be able to extract total DCR and TCR sequences from RNA-seq and single-cell RNA-seq data. So contributions and suggestions are always welcome. Here is also the link to the bioRxiv preprint that was just released last week, in case you want to know more details, and also the link to the NF Core Airflow website for the full documentation and features, and the link to the NF Core Slack in case you have any questions or requests or suggestions. We're very much open to communication there. So with this brief introduction of Airflow, I'd like to wrap up with this welcome talk, and we can move forward to the speaker talks.